Hello everyone. So welcome back to the NEET Success Simplified channel, and today in this channel we will be practicing lots and lots of questions from the chapter cell cycle. Yes, you heard it right. Whatever the possible questions which has came in the earlier NEET examinations, which can come, all those questions we will be dealing it with today. And if you have not watched the one shot session of this cell cycle, after this class, go back to the video, go back to the playlist and start watching that thing. Now, before that, before starting, I would also like to inform you that tomorrow at 4 p.m. I will be publishing a video which is going to consist of just 10 minutes, and in that 10 minutes we will be completing the whole cell cycle with the help of the mind maps. So now get ready and start with keep your pen and pencil and a notebook in your hand, and let's solve these questions. So the very first question today we have with is which says histone protein is synthesized in. So histone protein is synthesized in which stage of the cell cycle? Okay. So the options are, one says telophase, the other option says S phase, the other one we have the G2 phase, and then we have the M phase, right? Okay, sir. So here we have first option which is telophase, then S phase, then G2 phase, then the M phase. Which one will be the correct answer? Let me know in the comment section. Yes, the correct answer for this question will be what? Will be the S phase. Why, sir? Because do you remember that we studied that the histone protein is what? Histone protein is synthesized in the S phase. And one more thing, one more hint we can have from here is that during the S phase only, DNA synthesis also takes place. Yes or no? Yes, sir. During the S phase, DNA synthesis also takes place. And this particular histone protein helps in what? This particular histone protein helps in the packaging of DNA. So if synthesis of DNA is happening, that means DNA has to be packed also. So that's why in S phase only histone protein also gets synthesized. That's very good. Now let us move on and let's move with this very big question. Okay, to solve these type of questions, to solve match the following questions, what you need to do is that stop reading the question, start with the option. This is how you are going to save the time. So option A says A2, oh sorry, A3. So let's see A3. S phase represents interval between mitosis. No, so this one is wrong. Next option, let's go with the option number 2. This option number 2 says A with 4. So in S phase DNA replication takes place. Yes sir, in this thing the DNA replication takes place. Okay, now we have got two options which have A4. Now let's go with B1. So B is what? B says G2 phase. So in G2 phase, if I talk about G2 phase is basically 2. So inactive phase, no, G2 phase is not an inactive phase. So if I talk about this option, so G2 phase represents proteins are synthesized. Yes. So the correct option will be option number 3. So this is how you have to solve this question. If you will read this thing again and again, like during the examination you're going to read the question, then read the answer, it will take lots and lots of time. So solve the questions by reading the options. Now next we have which stage of meiotic prophase. See, the question has said which stage of meiotic prophase shows terminalization of chiasmata. Remember I told you that from this point the questions can be asked, and yes, this question was asked in NEET 2021. So terminalization of chiasmata takes place in which of the following stage? Liptotene, zygotene, fecitine, diplotene or dikinesis. So there was what? There was these stages: liptotene, zygotene, fecitine, diplotene and dikinesis. Okay, so in liptotene we saw that what? In liptotene we saw that thinnest chromosome, zygotene bivalent, trivalent, right? Fecitine crossing over, and in dikinesis chiasmata formation had taken place. So our answer will be what? Option number 3, which is dikinesis. Clear to everyone? That's great. Now let us move on with the next question. So the next question here says that which of the following stages of meiosis involves division of centromere? Sir, what was centromere? You remembered that if I'm talking about a chromosome, so this is what this particular structure is, the centromere. Yes, great. Centromere. So now our question is asking that which of the following stages of meiosis involves division of centromeres? Okay, metaphase 1 is in metaphase the division of centromere occurs? No, sir. In metaphase 1 there is no division of centromere. Rather, in metaphase 1 what happens? The chromosomes align themselves in the equatorial plate, right? So here the chromosomes align themselves to the equatorial plate. Yes. Now metaphase 2 also no division of chromatids takes place. Same on anaphase 2. What about the anaphase 2, sir? What about the anaphase 2? So remember, during the anaphase 1 we had this type of we had the homologous chromosomes, right? So the chromosomes were like this, they were attached, and in anaphase 1 homologous chromosomes were separated, right? And when it came to the anaphase 2, so in anaphase 2 what happened? The chromatids, the centromeres were broken. So in which stage the centromere is going to break? The centromere is going to break at the stage called the anaphase 2. Is it clear to everyone? Yes or no? Great, very good. So you understood how we are doing the questions. This way you have to approach the question in the similar manner in which we are doing it right now. Now let's move on and let's have this particular question which says the centriole undergoes duplication during. This is a very easy peasy question. It is saying that the centromere duplicates in which stage? So the centromere here duplicates in the S phase, prophase, metaphase or G2 phase. Sir, the centromere duplicates in the S phase only. Is that so? Is that so? The centromere duplicates in the S phase. Yes. In the S phase only centromere duplicates, DNA duplication takes place, and there is the formation of the histone protein. Clear? While in prophase, we saw that nuclear membrane got disappear, the nuclear envelope, and along with some of the cell organelles like the mitochondria, all of those things gets disappear during the S phase. Clear? Yes, sir, that is very much clear. Now let's have the metaphase. In metaphase, the characteristic feature is what? In the characteristic feature for the metaphase is the equatorial plate. Yes or no? That's very good. Equatorial plate is formed. In G2 phase, there is some proteins, except histone proteins. Some of the proteins are formed in the G2 phase. Is it clear to everyone? Yes or no? Is this question clear? That's great. If this is clear, it's very good. Now, let us move on with the next question. This question is very important and it is a good question. So before solving it, make it a star mark. Why? Whenever you are going to revise the cell cycle, whenever you are going to revise the questions, make sure to practice this type of questions. In this type of questions, you will feel like the answer is something, but the actual answer is something else. So let's quickly solve this question. The question says that the fruit fly has 8 chromosomes. Okay, great, sir. We understood that fruit fly is having 8 chromosomes. Okay. Now, it also said that this fruit fly is what? This fruit fly is diploid. This is diploid, which is 2n. Clear? Okay. Before moving forward, let us do one thing. Let's find the value of the n here. So, value of n will be 4. Means haploid content of this fruit fly will be 4. Great. Okay. Now, what the question is saying? During the interphase of mitosis, they are talking about the mitosis. Okay. They are talking about the mitosis. That means, are they talking about the gamete formation? No. That means we don't have to go into the meiosis stuff, right? Just the mitosis. So, during the interphase of mitosis, if the number of chromosome at G1 phase is 8, remember, they are talking about the number of chromosomes. What will be the number of chromosomes after S phase? After S phase. So, the answer will be what? Will it be 8? Will it be 16? Will it be 4? Will it be 32? Just remember a concept that DNA duplicates during the S phase. I am giving a very big hint. DNA duplicates during the S phase. DNA chromosomes number remains same. So there will be no change in the chromosome number. The chromosomes will have developed an extra chromatids due to the duplication. Suppose that before the S phase the chromosomes were like this. After the S phase the chromosomes will be like this. But still it is what? It is 2n, 2n only. Yes or no? Is it clear? There is no change in the number of chromosomes. There will be change in the content. So content will be doubled, but number of chromosomes will be same. And in the question, they are asking about the number of chromosomes. So the answer will be 8. I hope that this thing is clear. And this question is really very important. This type of questions are those questions in which many of the students do the mistake. They will take 16. And this is the question which is going to give you the rank in the NEET. Now, let's move on with the other questions. In case you have any doubts, let me know in the comment section. Okay, great. Now, let's move on and go to the next question. So, the next question here is what? The next question here says that dissolution of synaptonymal complex. Dissolution of synaptonymal complex occurs during. This was the concept which we discussed in the class. You remembered anyone? Anyone, do you remember? This was the same concept which we discussed. So it says that dissolution of synaptonymal complex occurs during liptotin, phecytin, zygotin or diplotin. Liptotin, phecytin, zygotin or diplotin. Which one should be? Come on, let's answer this question in the comment section. I want you to answer this question in the comment section. Anyone? Just remember, now we have what? We have the liptotin, thinnest chromosomes, right? Zygotin, formation, then phecytin, crossing over. Then we had what? Then we had the diplotin and chiasmata formation, dikinesis, terminalization. So what will happen in this thing? What is gonna happen in this thing? I'm waiting for the answers. Anyone in the comment section? Anyone in the comment section? Yes. Dissolution happens where? Dissolution happens in the diplotin stage. Dissolution happens where? In the diplotin stage. Yes. During this stage only dissolution of synaptonymal complex takes place. It's very simple. See how to remember. Liptotin, thinnest chromosome. Liptotin, zygotin was what? Zygotine, where all those things were happening, synoptimal complex were forming, fecitine crossing over. After fecitine, where it comes the diplotin, and in diplotin only dissolution will takes place. Clear to everyone? That's very good. Now let us move on with the next question. It says that some dividing cells exit the cell cycle. Okay, sir. Some dividing cells exit the cell cycle and enter a vegetative inactive stage. Vegetative inactive stage. This is called quiescent stage. This process occurs at the end of. Okay. I would like to know you about this inactive stage or the vegetative stage. So if I say that, see here, I am giving you one extra question here. I am giving you another question from here only. If I talk about that G0, in G0, cell is metabolically inactive. Cell is metabolically inactive. If I am saying that in G0, cell is metabolically inactive, is it correct or wrong? If I am saying in G0, cell is metabolically inactive, is it correct or wrong? So this statement is what? This statement is wrong. Cell is what? Not performing the reproduction. Cell is just not doing the reproduction. Else everything is done by the cell. Only reproduction is not performed by the cell. Rest each and everything cell is doing. Now, this quiescent stage or the G0 stage comes after which? This process occurs at the end of what? At the end of what? So if I say the cell cycle, we had what? We had G1 phase, we had S phase, we had G2 phase, and we had M phase. And after the M phase, we had what? We had the G0 phase. So we can write here, the answer is what? The answer is the M phase. Just remember that this G0 phase is not the part of the cell cycle. It exists the cell cycle, okay? It is existing the cell cycle. So this is what? This is the G0 phase. Clear? Okay, great. The answer is M phase. G0 phase comes after the M phase. Now, let's move on and let us have the another question. And this question says that identify the correct statement. Identify the correct statement with regard to G1 phase. Identify the correct statement with regard to G1 phase of interphase. Obviously, G1 is the part of interphase only. So here it says nuclear division takes place in G1 phase. Does nuclear division takes place? No. There is no division. There is no division. Interphase is what? Interphase is the preparatory phase. Interphase is what? It's the preparatory phase. Now next what we have? DNA synthesis or replication takes place. Is it G1? In interphase, DNA synthesis and replication takes place. But is that G1? No. It takes place in what? It takes place in S phase. Yes or no? Yes. Now, reorganization of cell components takes place. Is that so? Is that so? Reorganization? No. It takes place in what? It takes place in telophase. So last one will be our answer. Cell is metabolically active but does not replicates its DNA. Yes, in G1 phase, the cell is doing everything. Cell is trying to grow, but it's not doing anything which is related for the reproduction. Clear? It is metabolically active, but it is not replicating. Clear to everyone? Yes or no? If it is clear, let us move forward for the next question. Let us move forward with the next question. Clear? Great. Now, the next question here do we have is what? Match the following. Again, I told you how to solve the match the following questions. So, for solving the match the following questions, read the options. So, option A1 says A2. Let's go. Zygotine 2. Chiasmatic formation. No, it's wrong. A3. Crossing over. Zygotine crossing over. No, it's wrong. A4. A4 synapses. Zygotine में synapses form होता है क्या? In zygotine is synapses formed? Yes sir. In zygotine synapses is formed, synaptonemal complex, bivalent, tetrad, everything is there. Na? See, do you remember this type of structure? The homologous chromosomes pair themselves and they make us complex, which is synaptonemal complex. This is only the synapse, right? So synapses is formed. So A4 will be the answer. See, now you don't have to worry about the other options. If suppose we had this option also containing A4, then we would have gone to the next option. But only in a single option, in the single thing, you are getting your correct answer. So don't waste your time on reading the match the following questions. Just jump directly to the options. Clear? That's great. Now let us move on and let's talk about the crossing over takes place between which chromatids and in which stage of the cell cycle. Crossing over takes place with which chromatids and which stage of the cell cycle. Okay, sir. So crossing over takes between non-sister chromatids, sister chromatids. First of all, let us figure this out. So if I talk about the, this is the one chromosome, okay, the chromosomes from the S phase, and it's homologous pair, okay, yes. Now crossing over takes place between these two, yes or no? And these two are what? They are non-sister chromatids. They are what? They are non-sister chromatids. Yes or no? Yes, sir. They are what? They are non-sister chromatids. Clear? Great. Now what, sir? Now we have the other one. So let's, okay, every option is having non-sister. Okay. So let's read that thing. Non-sister chromatids of non-sister chromatids of non-homologous chromosomes. No, this is wrong. They both are what? They both are homologous chromosomes. They both are what? They are homologous chromosome. They are what? Homologous chromosome. Yes or no? So the first option is saying that non-sister chromatids of non-homologous chromosomes. So this is wrong. Non-sister chromatids of homologous chromosomes at Pectine stage of prophase 1. Is this statement correct? Think about it. Non-sister chromatids of homologous chromosome at Pectine stage. It's saying that Pectine stage. Crossing over takes place in what? It takes place in the Pectine stage only. So this statement is correct. Let's move on and let's read the other two statements also. It says non-sister chromatids of homologous zygotine. No. Fourth, non-sister chromatids of non-homologous, again wrong. So the correct answer here is option number 2. If you need the PDF of this particular questions, let me know in the comment section. Now, next we have this question which says after meiosis, it says what? After meiosis 1, the resultant daughter cells have. They are talking about meiosis 1. Meiosis 1. They have the same amount of DNA? No. Meiosis 1 is the reductional division. Twice the amount of DNA in comparison to the haploid gamete. Okay, they are talking about the haploid gamete. So, twice the amount of DNA in comparison to the haploid gamete. And the second, same amount of DNA in third will be same amount of DNA in comparison to the haploid gamete, and the fourth one is 4 times as compared to the haploid gamete. Okay, now see what they are trying to speak with us, that twice the amount of DNA. They are talking about the amount of DNA. Okay, so in meiosis 1, what happened? See, let's get back to the anaphase. So in anaphase, this was the condition. Okay, and during this anaphase, these homologous chromosomes got separated. This homologous chromosomes got separated. Let me draw a beautiful diagram for you all. So this homologous chromosomes got separated. One is this and one in this pole. Okay. And in telophase, they developed a cell membrane and cytokinesis took place. But if I talk about in gamete, they are having what? They are having this much chromosome, right? Only this much. So if I compare, if I suppose that I take this as C, so According to the gamete, the amount of DNA at the end of the meiosis 1 is 2C. Yes or no? So that's why option 2 will be the correct answer, and that is the reason why option 2 will be the correct answer. Clear to everyone? That's very good. Now let's move on and let us talk about that the cells in G0 phase. Again the same question. See, these all are the questions which are from the previous years. They all are the questions which has came in the previous year things. So see how well the questions are. Like in one they asked about the G0 stage, again they are asking about the G0 stage, right? So in G0 stage, cells in G0 phase exit the cell cycle, enter the cell cycle, suspend the cell cycle, or terminate the cell cycle. Which will be the correct answer? Which will be the correct answer? See, it's very confusing. Sir, you told exit kar jata hai, okay. You enter to nahi karega, that's very good. Second option can't be the thing. Suspend, terminate. So we are being confused this and this, right? Three options is creating confusion for us. Three options is creating confusion for us. Yes. So which one will be the correct answer? See, exit the cell cycle. Okay, this can be the answer. Suspend, this is more accurate. Terminate, it's not terminating. That means what? See, if that particular cell which is in G0 can divide the cell, right? Can start dividing when needed. So that's why we are going to say as suspend. There are some cells, I know, there are some cells like nerve cells which never divides. They are just sitting in the G0 phase. But there are some differentiated cells which has what? Which has exit from the cell cycle, but still they have the potential of division. So I can say suspend the cell cycle. We should not keep here as exit the cell cycle, okay? Because some of the cells having G0 stage, they have the ability to redivide it again. So we are making a generalized concept here, and according to that generalized concept, we are giving the answer. Clear to everyone? That's very good. Now let's move on and let's talk about this thing, the correct sequence of phases. The correct sequence of phases of life cycle, sorry, cell cycle. The current sequence of the phases of cell cycle. What is it, sir? It is M phase. No, M phase. No, M G1 G2 S. This is wrong. This is wrong. G1 G2 S M. Yes, this is correct. Is it? G1. Oh, sorry. This is also wrong. What is this behavior, sir? It's G2 here. This one is the correct option. G1S G2M. The correct option is G1S G2M. Clear? Great. Now let's move on with the next question. The next question here do we have, which says the stage which separating the paired homologous chromosome begins in the stage during which separation of paired homologous chromosomes begins in. Okay, one more thing, sir. I might get confused with the anaphase, but anaphase, they haven't given the option. Here they are talking about what? Here they are talking about the synapses or dissolution of the synaptonemal complex. How, sir? See the options, right? So the stage during which the separation of paired homologous chromosomes begins in. We just now discussed this question and the answer was diplotin. Yes, dissolution of synaptonemal complex takes place in diplotin. That means these homologous pairs are getting separated in diplotin stage. Yes or no? Clear to everyone? That's very good. Now next we have anaphase promoting complex, which is APC. Anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in human cells, which of the following is expected to occur? They are talking about the mitosis, okay? They are talking about the mitosis. So in mitosis, if suppose In anaphase, what is happening? In anaphase, the chromosomes are separating. Yes, yes or no? The chromosomes are separating. Let's suppose we have this one. Okay, we have this thing in mitosis, and what's gonna happen? This is, they are going to separate with each other, right? Yes, great. So what will not happen? Chromosomes will not be segregated. If this protein is not activated, if this factor is not activated, the chromosomes will not get separated. Okay, fragmented, no, the chromosome will not get fragmented. They're not cutting now, they're not cutting the chromosomes. Recombination of chromosomes arms will occur. This is all, this will also, this, there is no point of this particular thing. Chromosomes will not condense, no, condensation and all happens in the prophase, right? So this, so option 2 is the correct answer here. Now let's move on and let's talk about which of the following options gives the correct sequence of event during the mitosis. Okay, mitosis, they are talking about the mitosis. Great. So condensation happens. Okay, very good. But see, every option is having the condensation as their first option, first stage. So we can't differentiate. Now let's go with the second option here. Second option says nuclear membrane disassembled. Okay, this looks fine. Crossing over, no, this is wrong. Arrangement at equator, this is wrong. Condensation, nuclear membrane disassembly, okay. Crossing over, no, this is also wrong. So the correct option will be option number 1. Clear? It, condensation will take place, then what? Then the nuclear membrane is gonna disappear, the cell organelles is gonna disappear, then metaphase will come, and after the metaphase, centromere division, then segregation will take place, and they were talking about the mitosis. So in mitosis, crossing over will not take place. So this is the wrong option. 2, 3 and 4 were the wrong, but the correct option here is the option number 1. Yes or no? Yes, sir. Now let's move on with the another question. So we have more, how many questions do we have? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 21 more questions. So let's quickly solve those questions. Okay, great. Again, match the following states. How we can solve this match the following questions? By reading the questions. By reading the questions. Great. So let's read the questions. Sorry, by reading the options. A2, it says A2. A2. Facite in terminalization of chiasmata. No, sir. A4. Facite in chromosomes lying at the equatorial plate. No, sir. This is also wrong. 3. A3. 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 Crossing over takes place. A3. Crossing over takes place. No, this is wrong. Facite in A3. Yes. Crossing over takes place. See, this will be the correct answer. A1, pairing of homologous chromosomes takes place. No. So the correct answer will be what? The correct answer will be A3. I think so that this thing is clear to everyone. Yes or no? I think so that this thing is clear to everyone. Now, let's move on and let us go with the next question. Let us go with the next question, sir. Okay. The next question here we have what? We have when cell has stalled DNA replication fork, which checkpoint should be predominantly activated? Which checkpoint should be predominantly activated? See, cell is what? Cell is prepared for division. Okay, cell is just now, right now they are about to divide. Just maybe in 2 seconds they are about to divide. So before that, the cell is in which phase? The cell is between G2 phase and the M phase. The cell is about to divide. That means it is about to enter into the M phase. So checkpoint will be what? Checkpoint will be G2 and M. The checkpoint will be what? G2 and M checkpoint. Great. Clear, sir. Okay. Now let's move on with the next question. During the cell growth, DNA, this is a very easy peasy question, and this also came in the NEET. This question came in NEET. Can you understand? Can you guess? This is such an easy question. This is such an easy question. So during cell growth, DNA synthesis takes place in G2 phase, M phase, S phase or G1 phase. Sir, S phase. Yes. The correct answer here is what? The correct answer here is the S phase. Clear to everyone? Great. Now, let's move on with the next question. The next question says, in meiosis, crossing over is initiated at, see, how many questions were there from crossing over, like 4 to 5 questions we have already done with the crossing over. So crossing over is a hot topic in this chapter. So crossing over is what? Crossing over takes place in fecitine, liptotine, zygotine, diplotin. Tomorrow at 4 p.m. I'll be going to launch a video in which I will be discussing what are the hot topics for this chapter, and within 10 minutes with the help of a beautiful mind maps we are going to revise this chapter. I would like you to request to save that video so that during your examination time within 10 minutes you can complete your cell cycle. Now let's get back to the question. In meiosis, crossing over is initiated at fecitine, liptotine, zygotine, diplotin. What can happen? See, they are talking about what? They are talking about the activation, right? They are talking about the activation. So in which thing it will be done? Fecitine. Sir, in fecitine crossing over takes place. In fecitine crossing over takes place. In liptotine, synaptonymal complex formation takes place. In zygotine, in liptotine, the thinnest chromosome is present. In zygotine, synaptonymal complex takes place. In diplotin, what happens? In diplotin, terminalization of chiasmata. So in fecitine is only the place where crossing over is taking place. Yes or no? That's very good. Now let's move on, and this is a very easy question. This question you can do it very easily. Spindle fibers attached on to, you remember the structure which we have drawn? The structure which we have drawn was like this. Yes or no? Yes. And this structure, the centromere contains this thing, and this was what? This was the kinetochore. And the kinetochore is the structure where spindle fiber attaches and they pull the chromosomes like this, right? So kinetochore of the chromosome. See, to confuse, they have given kinetosome, and the students which are in hurry, they will see, okay, this, they can mark this also. So while attempting the question, be very sure that this is not kinetochore, it is kinetosome, which I think so never exists in the part of the centromere. Clear? Great. Now let's move on. Arrange the following events of meiosis in the current sequence. Okay, this question, if there is certain arrangement type of questions, what we do is that we run and start reading the questions. No, that will waste your time. See, these questions are very easy, but they are given in the NEET just because to waste your time. And if you are going to get trapped in that particular thing, you will lose your time and you will not be able to solve physics and chemistry, right? So start with the options. Best thing is start with the options. Okay, sir. So 1B, synapses. Synapses takes place first? No. Is it? Synapses takes place first? Yes, in this case, synapses is gonna take place first. Okay, after that we have the C, terminalization of chiasmata. No, without crossing over, chiasmata can't be done. So this option is wrong. Let's go with the option number 2. Synapses. And then A, crossing over. Okay, sir. After disappearance of nucleolus. And then we have what? We have the option number C. So, sir, disappearance of nucleolus occurs first, or the terminalization of chiasmata occurs. Here we will be having a confusion. So, yes, which is going to happen? Disappearance of nucleolus first or terminalization of chiasmata? Let me know. Let me know in the comment section. See, disappearance of nucleus occurs when? It occurs during the end of the prophase 1. Yes, disappearance of nucleolus, disappearance of nuclear membrane, everything occurs at the end of the prophase 1. But terminalization of chiasmata doesn't occur at the end. It occurs what? It occurs in the diplotin stage, right? So first terminalization will occur and then nucleolus disappearance will occur. Okay, so this is how we can figure it out, which is the correct answer for us. So option number 3 here is the correct answer. Clear to everyone why we chose? Because disappearance occurs at the end of the prophase 1 and terminalization is occurring inside, like in the prophase 1. So we have liptotin, zygotin, diplotin, liptotin, zygotin, fecitine, diplotin and dikinesis. So diplotin is there, after that dikinesis is there, and then prophase 1 is gonna end. So first of all, that terminalization of chiasmata will occur, then nucleolus will disappear. I hope that you have understood this question. If not, let me know in the comment section. I'll explain it again. Again, we have a very good question here, that a somatic cell that has just completed the S phase. Just they have completed the S phase, okay, of its cell cycle as compared to the gamete of the same species has. Okay, what they are saying, let's suppose that we are talking about our chromosomes. So we are 2n, okay. During the S phase, what is gonna happen? Our DNA will become 4C. Our DNA will become 4C. Yes or no? We have 2C and now it will become 4C. Sir, not able to understand 2C, 4C. See, during what? Like this we have, okay, we have this homologous chromosome, one from mother, one to father, one from father. Okay, sir. Now what, sir? Now here we have in S phase DNA duplication takes place. Okay, sir. So DNA will be duplicated like this. Yes or no? DNA will be duplicated like this. Yes or no? Yes, sir. So if we consider it as C, if we can, this is what? This is for us. This one is 2n. Yes or no? We are diploid. Yes, sir. Now see. This is a very easy concept which I am going to tell you. This is what? This is 2n and let's suppose this was C, this was C. So before the S phase, we were what? We were 2n, 2C. Clear? Okay, sir. Now what? After the S phase, what happens? This is N, this both is what? This both is 2n. But can you see? This is C, this is C, this is C, this is C. So in total, how many content we had? In total, we had 4C content. We had 4C content. Clear to everyone? So this is how from 2C we are getting 4C, right? But still, our chromosome number is still what? It's still 2n. So now we have same number of chromosomes, but twice the amount of DNA. Same number of chromosomes, but twice the amount of DNA. 2C, 4C. Yes, this can be the answer. Twice the number of chromosomes, no, there is no change in number of chromosomes. 4 times the number, no chromosomes change. Twice the number of chromosomes, no. So the correct answer will be what? Same number of chromosomes, but twice the amount of DNA. I hope that this thing is clear to you. Now, let's move on with the next question. The enzyme recombinase is required at which stage of meiosis? Which stage of meiosis the enzyme recombinase is required? This is a very easy question. This is a very easy question. In which stage the enzyme recombinase is required? See, we have fecitine, we have zygotin, we have liptotin, zygotin, fecitine, diplotin and dikinesis. In which stage crossing over takes place? Sir, crossing over takes place in the fecitine stage. And if the crossing over is taking place in the fecitine stage, recombinase enzyme will be required. So the answer will be what? Fecitine, option number 1. Clear to everyone? Great. Now, let's move on. Let's move on with the next question, which says in S phase of the cell cycle, amount of DNA doubles in each cell, amount of DNA remains same in each cells, chromosome number increased, amount of DNA is reduced to half. So in S phase DNA duplication takes place. Yes sir, in S phase DNA duplication takes place. So half to nahi hoga, half to nahi hoga. Chromosome number is increased. Sir, we have seen with the help of this explanation, we have seen that chromosome number remains same. So chromosome number will also not change. Will not change. Where it is? So chromosome number is not increased. Now second option, amount of DNA remains same. No, duplication हो रहा है तो amount of DNA can't remain same. This is also wrong. Amount of DNA doubles in each cell. So correct option will be option number 1. Okay, if you are liking this session, I would like you to request like, share and subscribe to this channel, and don't forget to watch a video which is going to come tomorrow at 4 p.m. in which we are going to revise cell cycle in just 10 minutes. Just 10 minutes. Now let's move on and let us discuss during which phase of cell cycle amount of DNA of cell remains at 4C level if the initial amount is denoted as 2C. This is also very easy. See, I'm going to explain you this thing with a very good example. You will never ever have the confusion whether when we say 4C, when we say 2C. Just concentrate, okay? So let's suppose your mother has this chromosome and your father has this chromosome, and this chromosome came into you. So this makes what? This makes 2N. This makes you 2N. For one chromosome, you have two things, right? Two copy of each chromosome. So this makes you diploid. Now what? Your cell enters into the S phase. Okay, sir. During the S phase, what is happening? DNA duplication is happening. Hi Tanish, how are you? During the S phase, what is happening? DNA duplication is happening. That means how DNA is duplicated? DNA is duplicated like this, sir. Okay. In the similar manner, this one also will have this type of duplication. Yes, sir. So now, what is C and what is 2C? See, earlier, before the S phase, you have this content C and C. One chromatid, one C. Okay, just remember, एक chromatid होगा, so you are going to say 1C. So here how many chromatids were there? 1C. Here how many chromatids were there? 1C. So in total you were having 2C of the DNA content. Okay. But after the S phase, what happened? See, your chromatid duplicated. So here you have what? C. Here you have C, and here you have C. And again you have this as C, this as C. So in this thing, how many chromatids are? Two chromatids. See, one this one this one. So total C and C, which will make 2C. Similarly, here also one chromatid, which is C and C, right? So in total, how many content? 4C. Yes or no? In total, how many content? 4C. So this is how 2C and 4C you can figure it out. Now let's get back to the question. So is it clear, Tanish? Did you understood about the concept of 2C and 4C? But see here, if I talk about the 2N, so it is 2N only. There is no change. Why, sir? Because unless and until the chromatids are not separated by its centromere, we are not going to say that it is N or 2N. Okay, if this chromatid will be separated like this, then we are going to say that the number of chromosome is increased. This chromosome is still one chromosome only. This chromosome is also a single chromosome trait till now. If they get separated, then we are going to say that they are now two different chromosomes. So that's why this is 2N only, but the content is double. Yes or no? That's very good. Now, what will be the answer to this question? The question says, during which phase of cell cycle amount of DNA is in cell remains at 4C level if the initial amount is 2C. So what is going to happen? See, during the S phase and after the S phase, this is happening. So in G1 phase, we will not have this one, right? G1 phase, the normal condition will be there, right? And then we have G2 phase, then we have G2 and M phase. So in both G2 and M phase, we will be having this kind of condition. So the answer will be G2 and M phase. Clear? Why, sir? Why we have not chosen option number 3? Because we had a better option here, which was also mentioning the M phase. If suppose this option was not there, then we would have chosen option number 3. But it is saying M phase also, so option number 4. Clear to everyone? That's very good. That's very good. Now, let us move on with the next question. And here, this question says the complex form. This is a very easy question. It was also asked in NEET. This is a very easy question. The complex formed by a pair of synapsed homologous chromosome is called. pair of synapsed homologous chromosomes. Synapses, remember synapses, where it was formed, sir? It was formed in the meiosis 1. In meiosis 1, prophase 1, we have liptotin, zygotine, fecitine, diplotin, dikinases. And in liptotin, the chromosomes were thinnest. And then in zygotine, those homologous chromosomes paired and form synaptonymal complex or tetrad or bivalent. Yes or no? What is this bivalent tetrad synaptonymal complex? We have discussed this thing in the class in the previous video, but I am gonna explain this thing very quickly. Okay, so during the meiosis 1 and during the meiosis 1, prophase 1, during the zygotine, what happens? Two homologous chromosomes pairs, and this forms what? They form the complex which is known as synaptonymal complex. But we can also say it as tetrad. Why? Tetrad is the term which is what? Thank you so much. Tetrad is what? Tetrad is basically the term which is given. Tetrad is basically the term which is given for the chromatids. Tetrad is term which is given for the chromatids. So how many chromatids? 1, 2, 3, 4 chromatids. And here if I talk about why bivalent, sir, why bivalent? So bivalent is given, the term bivalent is given to the, with respect to the number of chromosomes. So can you see here how many number of chromosomes do we have? We have 2, so that's why bivalent. Clear? If in the options, let's suppose tetrad was also given, so tetrad can be also the correct answer. That's very good. That's very good. I hope that you all have understood this question. If yes, let me know in the comment section. Now, let us move on with the next question. Okay. The next question says that one more thing. These are the questions which are very texty. And in NEET, if you are going to read these questions again and again, it will be time waste for you. So make sure that whenever these texty questions come, you have to read it only once and then solve the questions. Okay, don't read again and again. So whenever you are reading this type of questions, your concentration should be on this question. Otherwise, it will waste your time. A stage in cell division shown in the figure. Okay, I'm so sorry. There is no figure. Okay, there is no figure, guys. So let's do one thing. Let's skip this question. In the next class, I'll bring this question and with the figure. Okay. Now, let's have identify the meiotic stage. Identify the meiotic stage in which the homologous chromosome separates. See, again the same thing. Again the same question is repeating. That means we have identified some hot topics in this chapter. One is, sir, what? One is the crossing over. Yes or no? Crossing over से questions आते हैं बहुत. Yes. Next what? Next hot topic is what? It is the chiasmata formation. Yes or no? Second hot topic was S phase. Fourth hot topic was what? Number of chromosomes. So these are what? These are the hot topics of this chapter. Majority of the questions comes in NEET from these chapters. Now, identify the stage in which homologous chromosomes separates while the sister chromatids remains associated. And this question is from anaphase thing. This question is from the anaphase thing. Yes or no? Yes, answer the question. I want the answer in the comment section. See, here the answer will be what? The answer will be anaphase 1. How, sir? How? See, during the meiosis 1, what happens? This is the homologous chromosome, yes or no? And this is what? This is separated during the anaphase 1. This is separated during the, oh my god, why I am not able to make this chromosome? Okay, so this is separated during the anaphase 1, and in anaphase 2, the sister chromatids are also separated. In anaphase 2, the sister chromatids is separated. So it is very easy. This question was very easy, right? It's just asking in which stage homologous chromosomes were separated. So homologous chromosomes were separated in anaphase 1. If they would have said in which stage the homologous chromosomes having the sister chromatids are separated, then it will be anaphase 2. Clear? Any doubts in this question? Tanish, review on point. Any doubts in this question? Let me know in the comment section very quickly. If you have any doubts, let me know in the comment section. Great. Let's move on. This is the question. This is the question. Given below is the diagram. Okay, I hope so that your diagram is visible to you. Given below is the representation of a certain event at a particular stage of type of cell division. Which stage is this? See this stage. See this stage. See this stage. What is happening, sir? Here, you can guess this thing from this particular thing. See, what is happening? Here, sir, crossing over is happening. Here, crossing over is happening. What is happening, sir? Crossing over. And if crossing over is happening, so in which stage crossing over takes place? Will it take place in prophase of mitosis? No, sir. Mitosis may no crossing over. Both prophase and metaphase of mitosis? No, sir. Again, mitosis. No need of thinking. Prophase 1 during meiosis, yes. So crossing over takes place. What? Crossing over takes place in the chitin of prophase 1. So option number 3 is the correct answer. Option number 3 is the correct answer. Clear to everyone? That's very good. Now let's move on with the next question. The next question says that during gamete formation, the enzyme recombinase participates during. Recombinase enzyme, again, recombinase enzyme, where it is required? It is required during the crossing over. It is required during the crossing over, yes or no? So crossing over, prophase 1. See, how many questions from crossing over has came from the past years? So crossing over these topics, screenshot these topics during your NEET preparation. Read these topics in much concentration, practice the questions from these topics, so that if you leave other things also during the last few days, it will not matter a lot. But these topics should be done 10 times, 20 times, 30 times, because they are the hot topics from where the question is coming. Yes or no? Great. Now, Tanish, in which class do you have? Are you in which class? Are you in 11th? Are you in 12th? Or are you a dropper? Which one? Now, next we have what? At metaphase, chromosomes are attached to the spindle fiber. Obviously, that is kinetochore. That is kinetochore. We discussed this question. Now, okay, this is a good question. This is a very good question. So, we have what? We have select the correct option with respect to mitosis. Okay, before this, I have a great news for you. Tomorrow at 4 p.m., I will be publishing a video which will be just of 10 minutes, and in that 10 minutes we will be completing the whole cell cycle with the help of the mind maps. So you need to save that video and revise again and again. See, revision is the key for success, and jitna kam time revision mein lagega, that is more beneficial for you, right? Okay, now let's get back to the question. That's very good, Tanish. So chromatids separate but remain in the center of the cell in anaphase. Select the correct option with respect to mitosis. So in mitosis, just imagine, chromatid separates. Okay, sir. Chromatid separate but remains in the center of the anaphase, center of the cell in anaphase. No, it's wrong. It will go into the poles, right? Chromatids start moving towards the opposite poles in telophase. This looks correct, but let's move on with the other options also. Golgi complex and endoplasmic reticulum still visible at the end of prophase. No, it's wrong. Chromosomes move to the spindle equator and gets aligned in the equatorial plate in metaphase. Equatorial plate in metaphase. Okay, sir, we had two options. This was looking correct and this was looking correct. But which one is more accurate? Which one is more correct? See, chromatid starts moving towards opposite pole in telophase. No, it starts moving in opposite poles in the anaphase only. Remember, separation hua and the chromatids will go into the pole. So they have started moving in anaphase only. In telophase, to cytokinesis and everything will happen, right? So this one is also wrong. The correct option will be option number 4. The correct option will be option number 4. That's great. Any doubts in this question? Let me know in the comment section. Any doubts in this question? Let me know in the comment section. No doubt? That's very good. I hope that you are liking this session, and in case of any doubts, you know what you have to do. Okay, so we have a diagram. Now we have a diagram here, and this diagram says what? This diagram says, okay, why my eraser is not working? Yeah, which stages of cell division do the following figures A and B represent respectively? This is a very easy question. I want this question. Everyone, I want this question to solve this. Everyone, please see this and solve this question. This is the diagram. Sir, the diagram is what? The diagram is representing some of the phases. And what is it? This looks like something, what? This looks like something anaphase, pulling off the chromatids to the pole by the spindle fibers. So A represents what? A represents some anaphase. Okay, so A represent anaphase, yes or no? That is late anaphase. And B represents what? B represents the prophase. Why prophase, sir? See, the nuclear membrane, the diagram is not little bit clear, but nuclear membrane is starting to dissolve. So that's why we can say it is prophase. Clear to everyone? That's very good. Now, let's move on with the next question. Let's move on with the next question. Now, we have the question which says how, okay, let's should we do a one question from the Mendel also? Should we do a one question from Mendel as well? I have one question from Mendel here. How many true breeding pea plant varieties did Mendel select as a pairs which were similar except in one character with contrasting traits? It's a very easy peasy question, sir. This is a very easy peasy question, and answer is 7. Answer is 7, right? Great. Now, let's move on with the another one, and the question says that during mitosis, ER and nucleolus begins to disappear at, this is also very easy, begins to disappear at early prophase, late prophase, early metaphase, late metaphase. Sir, we know prophase is the correct answer. Like for us, we know that prophase can be the correct answer, but here in options they have given early and late. Early and late. So see, at the end of the prophase, everything is disappeared. At the end of the prophase, everything is disappeared. It's starting, right? So starting will be in the early prophase. Starting will be early prophase. That's very good. Now, let's move on. We have 2 or 3 more questions. Synapses occurs between, again, question from the crossing over. Synapses occurs between 2 homologous chromosomes, a male and a female gamete, mRNA and ribosomes, spindle fibers and centromere. No, mRNA, ribosomes, spindle fibers, centromere, all are these shitty options. This is also male and female gamete में fusion होगा. Synapses क्या करेगा? So this one, 2 homologous chromosomes, right? And it happens where? It happens in the zygotine stage. It happens in the zygotine stage. Great? Yes. So this was all the PYQs questions which we have discussed today, and what you have to do is now you just have to see that how many questions you were able to solve and how many questions you were not able to solve. The questions which you are not able to solve, what you have to do is that you have to make a note of it and search that the question was from which topic, write it down and revise those topics. Till then, bye-bye everyone, and let's meet tomorrow at 4 p.m. for the great revision thing. See you in the next class everyone. Thank you. Bye-bye.